thank you then. Hi, my name is Victoria Schmalhofer, and I will be discussing interaction modifications. When looking at species interactions out in nature, the classical approach in field ecology has been to focus on pairwise interactions. But in recent decades, it's been recognized that there are limitations to this approach because species don't occur in pairs in isolation in nature. The presence of other species is very likely to lead to indirect effects. And in recent decades, there has been a lot of interest in what the impacts of multiple species in the community will be. There are two broad categories of indirect effects that have been examined, and these are interaction chains and interaction modifications. In an interaction chain, a series of direct interactions makes it possible for one species to indirectly affect a third through a shared intermediate. So for example, if species A eats species B and B eats C, then A has the potential to indirectly and most likely positively exert an impact on C through that shared intermediate B. Interaction chains can be predicted from the direct interactions amongst members of the chain and are quite straightforward. Interaction modifications, on the other hand, are not predictable based on the direct interactions of species pairs. And this is because rather than directly interacting with another species, one may exert its effects by affecting the relationship between two others. So for example, if species A, rather than physically directly interacting with B, if it somehow changes B's behavior, and that behavioral change affects the relationship between B and C, then A can be having an indirect effect on C. It's thought that interaction modifications are quite common in natural systems. However, experimental manipulation is required to uncover their presence. And flowerhead communities offer an ideal system for investigating the potential of interaction modifications. And this is because in a typical plant community, you have a wide variety of plant species in bloom at any given time. These plants will support a variety of pollinator species, and these pollinators will in turn support a variety of predators, spiders among them. And it is highly unlikely that insects will be responding in the exact same way to the various plant species available to them. And so these differential pollinator responses may affect predators using the various available host plants. For a number of years now, I've been investigating interactions between a flower-dwelling predator, its host plants, and the suite of pollinators that utilize those same host plants. Earlier research showed that when spiders were using bidens as a host plant, they experienced better foraging success than did spiders that were using concurrently blooming solidago as a host plant. And this gives rise to an interesting question. From the spider's perspective, is solidago always an inferior host, or was solidago an inferior host because of bidens? So if we removed flowering bidens from the system, would foraging success of spiders on solidago improve? In other words, is an interaction modification occurring? So this was what I set out to examine. Now, for those of you not familiar with the characters in this little ecological drama, let me introduce the players. My predator of focus is Mizuminoides formosipes, the white-banded crab spider. This is a relatively common flower-dwelling crab spider, and it is preying primarily on pollinators. Adult female spiders can take quite large prey, up to and including honeybees and bumblebees. And they employ the standard crab spider hunting method, where a spider sits and waits for potential prey to essentially walk into its embrace, at which point it uses its enlarged raptorial forelimbs to grapple the prey, hold it, and then deliver an envenomating bite. Adult female spiders gain most of their mass during that adult stage, and this has crucial impacts on their eventual fitness. Physiological changes associated with insemination and egg maturation, as well as hunger level, could influence predatory motivation. So I had a protocol to try to standardize this. I collected up my spiders as penultimate instar juveniles, brought them into the lab, and ran them through a time frame where I controlled their diet and I gave them mating opportunities so that I could try to ensure as best I possibly could that when I released them into the field, all spiders were equally motivated to hunt. Now, having a look at the host plants involved here, Solidago juncea, otherwise known as early goldenrod, has a flower head that is described as a compound panicle. It's composed of hundreds of tiny little individual inflorescences, which are arranged in these elongate panicle branches, and then multiple panicle branches together compose the compound flower head. It's very complex, very three-dimensional. Now, a floral unit on this plant is the area of the plant that is occupied by the spider. It's used by the spider as its hunting arena, and it's also functioning as a landing platform for visiting insects. And so on Solidago, the floral unit is one of these panicle branches. The floral architecture of Bidens aristosa, commonly known as beggarticks, is quite a bit different. It's much more two-dimensional, and here the floral unit is a single inflorescence, which is quite large, and is composed of a central disc of small disc florets that are surrounded by 8 to 10 large petal-like ray florets. My experiments took place at the Hutchinson Memorial Forest Research Area, which is located in central New Jersey. And my field site was a 1 hectare old field that was dominated by foxtail grasses. I took a small section of the field, mowed down the grass, and established a grid within this mown area. So on my 10-meter square grid, I had randomly arranged 4 patches of Bidens and 4 patches of Solidago. Each patch was about 45 flowering stems, and patch placement remained consistent throughout the duration of the experiments, which took place as a series of 3 separate runs, which was done to accommodate the flowering phenology of the plants. In each run, both plant species are present, but both are not necessarily flowering. So this holds the vegetative background constant. The only thing that's being varied is the floral signal that pollinators are receiving. So in the first one, Solidago is in bloom, Bidens is present, but not in bloom. The plants are still just in bud. During the second run, both plant species are flowering, and in the third, Bidens is in bloom, but the Solidago flower heads have senesced at this point, so they are effectively no longer blooming. A given run lasted for 3 days, and over a 10-hour diurnal observation period each day, I visited my patches at regular intervals to collect my data. 4 spiders, each of which was individually marked with a number on the abdominal dorsum. 5 were released into each patch at the start of a run. I recorded their activity. Were they hunting? Were they feeding? If they were feeding, what were they feeding on? Where were they located? No caging or tethering was used, so spiders were free to move within their patches or to move between patches. And each spider was used in only a single run. I also had data on spider size. Their mass was determined prior to release and after the run was done. I calculated body condition, and there were no significant differences in size or body condition of the spiders that were randomly assigned to the various experimental treatments. Some migration did take place, and I had a protocol in place to handle that when it occurred, and I'd be happy to discuss that later. In terms of the insects, at these observation intervals, I was recording number and identity of the insects that were visiting the patches. Data were analyzed using multivariate analysis of variance with associated ANOVAs. I transformed the data as necessary to meet test assumptions, but for this talk, I'll be presenting raw data in all my graphs. Just to remind you, the primary comparison of interest here is what is happening on Solidago host plants in terms of spider foraging success and pollinator responses in the presence and absence of flowering Bidens. Of secondary interest is when Bidens is not present, is Solidago just as good a host plant as Bidens? So does what happens on Solidago host plants in the absence of Bidens differ from what's happening on Bidens? So let's have a look at the data for the spiders. First off, the experimental treatments did in fact have a significant impact on the 3 measures of spider foraging success that I used, both rate of mass gain, prey capture rate, and size of prey captured. All 3 of these varied in a similar manner, so I'm going to focus on rate of mass gain. And initially here, focusing on just what is happening on those Solidago host plants, what we see is that in the presence of blooming Bidens, there is a significant reduction in the rate of mass gain of spiders that use Solidago as a host. And in fact, their rate of mass gain is about half of that of spiders on Solidago hosts when Solidago is alone. Comparing how spiders are doing on Solidago alone versus how they do on Bidens, we see that there is no significant difference in the spider's rate of mass gain amongst these 3. This is evidence for an interaction modification in this system. And this is just to show you that the rate of prey capture and size of prey capture are showing similar results where in the absence of bidens, spiders do equally well on solidago, but once you bring flowering bidens into the system, the foraging success of the spiders on the solidago hosts decreased significantly.
It's been 10 minutes.
Thank you. Now, what's going on with the insects? Again, we see that the experimental treatments are having a significant impact on pollinator responses in terms of the size of the visitors and their rate of visitation. And let's focus on visitor size first. Again, looking at that primary comparison, what's going on solidago. Here we see that in the presence of flowering bidens, there's a significant reduction in the size of the average visitor to the solidago host plants. Average visitor size is about half that you see on solidago when solidago is alone. Comparing solidago alone against the bidens host plants, there's no difference in the size of the average visitor to these plants. So the presence of flowering bidens is drawing larger pollinators away from those solidago host plants, and they're shifting over to bidens. Results are similar for insect visitation rates here on the left, with a significant decrease in the number of visits per stem per hour to solidago hosts when flowering bidens is present. Otherwise, solidago is attracting equivalent numbers of insects compared to bidens. Visitation rates per floral unit are a little bit more difficult to interpret, and this is because there was a big difference in the number of inflorescences per stem between the second and third runs that I was doing. The numbers here in white are telling you the average number of floral units per stem. And again, I would be happy to discuss that later, but I just want to wrap up here by saying, yes, indeed, there is an interaction modification taking place in this system. In the presence of flowering bidens, the foraging success of spiders on solidago host plants is reduced, but in the absence of flowering bidens, solidago is a perfectly suitable host plant for the spider. This interaction modification is being driven by changes in pollinator behavior. Basically, when flowering bidens is available, the insects shift from solidago over to bidens. The shift is particularly pronounced in Hymenoptera, especially the larger taxa such as bumblebees and honeybees, which are the most profitable prey items for these spiders, and the honeybees are a definite preferred prey item. And this may have very, very significant impacts on eventual spider fitness, and that is something to be looked at a later point. At this moment, I would like to acknowledge my sources of funding for doing this research, and I would be happy to answer any questions that anyone might have.
Thank you, Victoria. Questions come in right away from Ann Ribstra, who said, do you have any evidence of frequency dependence? And did you look at a gradient of plant density?
Okay, in this particular study, I did not look at a gradient of plant density. I was trying to keep my densities as consistent as possible between my different plant species. Okay, please elaborate on the question of frequency dependence.
Yeah, Anne, I think she wants to know if you mean frequency dependence of, she says for the plant's relative density of each species.
Okay, I'm still not following.
Okay, I'm gonna let Anne maybe, she says just go on. I'm gonna let her contact you directly. Sure. The next question comes from Andrada Opris, who says, why did the insects prefer bidens as well, structure-wise?
Okay, let me flip over here. Okay, this is the one I want. Okay, when you compare these two plants, bidens is throwing a much, much larger and stronger floral signal. So because the individual inflorescences are much larger, both of these plants have an ultraviolet signal. So each of them is one of these central discs surrounded by ray florets. But on solidago, the individual inflorescences are so small that it all kind of blurs together in a mosaic. In the UV, that central disc and the proximal portion of the ray florets right around the disc absorbs UV, and the distal ends of the ray florets reflects. So you get a very strongly contrasting UV signal. It's much more obvious in Biden's because Biden's is so much bigger. You can see the comparison of inflorescence sizes here. Biden's also has the disc florets are larger and deeper, and so there is likely a much greater nectar volume here. So it may be a more profitable plant for the insects to be feeding on in terms of more bang for your buck for each disc floret probed for nectar. So I think that's what's going on there.
Yeah, I was guessing there was probably a UV situation that was making these things more attractive. Victoria, thank you so much for the talk. You've got more questions in the chat. Okay. So if you can go ahead and address those there.
Okay, and I will stop my share.